what happens when the going gets tough? The tough get going. So when bacteria are in trouble, they grow these electrical motors and flagella, and they start spinning them and swimming away. But where? Where to swim is the question, right? We all need an internal guide. Let's take a nice deep sigh of relief. Welcome. To tell us where to go. So bacteria have inside them a little computer made of 6 proteins that can tell them where to go. And we can all learn some valuable lessons from this computer. This is called bacterial chemotaxis. In this lesson, we are going to talk about bacterial chemotaxis and use it as an example for the principle of robustness. Principle of robustness that we started in the last lecture. So I guess. Principle of robustness. Biological circuits are designed so that their essential function is almost independent on the naturally varying parameters. We need to know what the essential function is and what those parameters are. And last time, remember, we talked about the input-output relationship of a signal transduction system and showed that most designs you can write down on paper make that input-output. Let's take a nice deep sigh of relief. Welcome. Shalom. That's why I take a nice deep sigh of relief, to welcome you. And not to, you guys can come in whenever you want. It is invariant, really mathematically invariant to changing the concentrations of any of the proteins in the circuit, the receptor and the transcription factor. Remember? Yes. So today we'll talk about a different essential function called exact adaptation in this particular circuit. All right. So we're done with the review. Let's talk about bacterial chemotaxis. So I drew here some cigar-shaped cells. Those are E. coli. And they have several, let's say 3, 4, 6, 10 flagella each. And they're swimming. They swim pretty fast. They swim 30 microns per second. Their size is 1 micron. Then they swim 30 times their body length per second. That's faster than you can run in proportion, right? And if I put a pipette here, and inside the pipette I put something delicious, a chemical, a attractant, like, for example, the amino acid aspartate, the bacteria will come and form a cloud here at the mouth of the pipette and go into the pipette. If you look in the microscope, this was discovered mid-1900s. And if I put there something that they don't like, a repellent, I don't know if it's with an E or an A, like for example, nickel ions or the amino acid leucine or some other stuff like that. Why leucine? Right? Let's take a nice deep sigh of relief. I'm glad you're attracted to the course to come back. They go away. They run away from this pipette. In other words, they can sense gradients of chemicals and climb up gradients of attractants and go down gradients of repellents. This is called bacterial chemotaxis. They go up gradients of attractant. Chemotaxis, when they sense a chemical, they call for taxi. The origin of the word. how do they do it? So in this class, we talk about how they move. Then we look inside the cell to see the circuit. Then we'll make mathematical models of the circuit to try to understand about robustness. That's our plan. They can sense an incredible dynamic range, so they can sense more than 5 orders of magnitude of gradient strength, and gradients as small as 1 molecule per bacterial length over a background of 1,000 molecules. So if I imagine around this E. coli volume, that's about the volume of E. coli, 1 micron cubed, and I put there 1,000 molecules per micron cubed, which is about 1 micromolar. And the gradient, this is so small that there's an average 1 molecule difference between the head and the tail of the bacteria, 1 nanometer per micron. They can detect it. Now that's incredible, because just the fluctuations are square root of the number of molecules. So 1,000 plus minus 30. That's the fluctuations. But they can still detect 1 molecule difference. And they're so small, experiments actually rule this out, that they can't really understand what's their head and their tail. They can't have 2 antennae there and measure the difference. And when you see them moving, they really don't keep one head in one direction, one tail in one direction. That's not the way they work. They're too small. And also, they're facing tremendous physical constraints. They're so tiny that Brownian motion is important. That is the number of water molecules. Let's take a nice deep sigh of relief. Hello. There's places over here if you want. Number of water molecules hitting them from the right or the left is different. And so if they try to swim 10 seconds in a certain direction, after 10 seconds on average, they find themselves 90 degrees away. It's like if you try to walk inside in a hurricane that carries you like that. But they can still climb up the gradients. So their dynamic range is very impressive in their sensitivity. Now, how they do this was discovered in the 1970s by Howard Berg. Howard Berg. He was a physicist, and he built a microscope that could follow the bacteria as they're swimming. And the miracle was that this keeps the bacteria inside the center of the field of view. And he did that with 1970s electronics, which had it was right at the cutting edge of technology back then. Sorry. And what he saw was this, that bacteria, the bacterial chemotaxis, bacteria perform a biased, random walk. And it looks like this. When you look at them, now there's no gradients or anything, just a homogeneous fluid. They swim straight for about a second. This is called a run. And then they tumble. They randomly change direction. That's called a tumble for about 0.1 seconds. And then they swim in a new direction. Tumble, swim, tumble, swim. The runs have exponentially distributed lengths with an average of about 1 second. They tumble about once per second. We can say their tumbling frequency is about 1 per second. This will be important, this word. Tumbling frequency means how many tumbles they make per second? About 1 tumble per second. There was a question.
When are they doing it? In what situation? Like what is going on in the environment?
What is going on in the environment in this experiment? It's a uniform liquid with no gradients. Did I answer your question? Mm-hmm. Okay. Now, just if I look a little bit closer at the bacterium, I notice that the motors connected to the flagella can turn counterclockwise or clockwise. So they're not only electrical motors that spin at 100 hertz, they're also bidirectional. When they spin counterclockwise, the flagella the proteins make the flagella have a helicity, and they like this counterclockwise direction. They go to form a bundle, and this gives you the run. But enough that one flagella, one motor flies counterclockwise, that disorients the bacteria. That makes flagella fly apart. Nice deep sigh of relief. And that makes a tumble. Yeah. So that's how the bacteria can switch between runs and tumbles. Now, how do they climb the gradient? It's very simple, what Howard Berg discovered. The bacteria climb the gradient by comparing the attractant concentration now with the attractant concentration in the past. And if things are getting better, attractant goes up, nice deep sigh of relief, then that means they think we must be going in the right direction. And they lower their tumbling frequency. So they keep going in the right direction. So if attractant increases in time, lower tumbling frequency, and keep going straight. And if the attractant concentration decreases, let's say now is less than ahead a little bit before, they increase the tumbling frequency. Now these tumbles are random changes of direction. So they have very little control over their destiny. But what I say, if things are getting worse, randomly change direction. And maybe I orient myself in the right direction, maybe, but maybe not, and then I change direction again. So this little statistical game is what makes them, helps them perform chemotaxis. So if I think of all these bacteria cloud, they're doing, if you look at them, even with the gradient, the first thing you see is that they make the same kind of random walk. But if you look a little bit closer, see that the center of mass of this entire cloud of bacteria is slowly moving up the gradient because of this bias. If they measure the attractant is increasing, they have less chance to tumble, and on average, they go up in the right direction. The typical velocity, if they're swimming at 30 micron per second, typically this cloud moves the center of mass moves much slower, let's say 1 micron per second, a few percent of their velocity, but still enough it's a little bit like electrons in an electric field or something. They still move up the gradient. Yes?
How dense are they? Are they colliding or not?
So how dense are the bacteria? Normally, in a liquid test tube, they're very far from each other. Only, let's say, 1 in 10,000 of the volume is made of bacteria, and they don't collide. But then sometimes they also live in our body in biofilms and surfaces where they're really, really dense. And then they have additional ways of moving. They differentiate into long cells that have about 100 flagella, and they start what's called swarming with the help of each other, and they secrete all kind of surfactants. Really amazing differentiation into different kind of species. But we're talking here, let's say, about a dilute situation. By the way, they can also secrete attractants and cause this kind of gravitational collapses where the bacteria fall. I say gravitational, of course, as an analogy. They form these amazing shapes on the agar plates that because they attract and repel each other and things like that. But we're not going to talk about that. So that's bacterial chemotaxis. Yeah?
Can they control the distribution, the probability distribution of the angle?
Can they control the probability distribution? Let's take a nice deep sigh of relief of the angles and the tumble. No, they can't. As far as detailed experiments, the angle is random. I mean, it's slightly biased in the forward direction, probably for mechanical reasons. It's not completely uniform, but they can't control it with relation to where they're pointing, because in reality, they don't know where they're pointing, and they can't know where they're pointing. All they can do is do a temporal comparison of now and the past. And you might ask, how can bacteria do that? This 1 micron thing. How can they have a memory? I mean, it's hard for me to have a memory. I don't remember names and stuff like that. How can the bacteria remember? And we'll talk about it, this kind of memory, how it biochemically works. All right. And the bacterial chemotaxis appears in most species of bacteria. So let's take a nice deep sigh of relief. Scare them away. No, just kidding. It's going to come back from the other side. So E. coli has a sense of smell. It can sense several dozens of chemicals at least. But other bacteria can sense light. That's called phototaxis. This escape from the light. Other bacteria can sense magnetic fields. They grow these little nanometer-sized magnets from magnetite crystals inside of them. And they use that to know where it's up and down, because the magnetic field of the Earth is the only sign they have in the mud, where it's up and where it's down. Electric fields, bacteria living in silver mines can chemotax towards silver. It's wild. Yeah.
Regarding the tumble frequency,
Yep.
they have to do, if they have to increase and decrease it, they have to do it each time they move, right? Because sometimes they can move to the right way, and sometimes they can move to the wrong way. like each move. So you have to transfer the information into the decision of changing the frequency in each move.
So the question is, in each move, they need to determine the tumbling frequency. We'll see. It's a differential equation. They do this continuously. It's not a discrete. It's not a discrete move, run, tumble. I mean, their motion is discrete.
But if the movement is like randomly, like to different angles, it can like if you, for example, twice he was going to do.
Okay, I'm going to stop you.
Mhm.
Wait and see if at the end of the class you have this question. Okay. Yeah.
Regarding the question about the angles, you said earlier that they have many models, not only one.
Right.
So basically, if each model has a separate statistics for tumble and run, they should have some bias according to the distribution of the model around
The comment is since they have many motors, maybe there's a bias in the angle.
if they're not distributed homogeneously on the shell.
Okay. Good point. More questions? Yeah.
Is this sort of tumble frequency decision apparatus the rule for everything that has a flagella, or do other like organisms that also have flagella move in different
Yeah.
ways?
So there are other ways to taxi. If you have flagella. And yeah, there are. For instance, you could imagine that you change your velocity according to not their. So there are organisms that do that, and then there's a whole range of strategies. But I think this one has been observed in a wide range of different species, and it maybe has some advantages. But there's other ways to do it too. There's all kind of twitching motility, gliding motility. I think most ways of motility in biology are still unknown. We don't. I mean, the community doesn't understand how they work, why they work that way, how the computer works. But bacterial chemotaxis, I would say, for me, it's a point of light in biology. It's a system that is, I love it like necrophilia because it's been studied to death. But that's the place where you know the molecular interactions enough to begin to make very accurate models and understand principles. So, and the mechanisms inside bacterial chemotaxis are quite universal. I mean, the same kinds of components are built, mixed and matched to make other kind of signaling systems. But there's a huge diversity of possible behaviors. This is found quite often. All right. Where are we? We took runs, tumbles, clockwise. OK, congratulations. We finished the first part, which is understanding the physiology of how they move. And now we're going to move into a, well, not yet. We still need to do a physiological experiment. I want to get quantitative. So here's the typical experiment done on bacterial chemotaxis. And the experiment is as follows, to give us some variables to play with. The experiment is, oh, by the way, if instead of attractant I put in repellent, it's exactly the opposite. Climbing up gradients of repellents, if repellent increases, you increase the tumbling frequency. Right, so here's the experiment. So in experiment, we add attractant uniformly in space. So I have them swimming, and now I mix very quickly a lot of attractant. There's no direction, just everywhere they're going, nice deep sigh of relief. Anywhere they're going, they see such conditions get better, no matter which direction they're going. Okay? And I'm going to plot what happens. So you observe the bacteria. This is time. And this is their tumbling frequency. And this is how much attractant. So I start with no attractant. Tumbling frequency is about 1 per second, like I said. Well, averaged over the cells. Now I add attractant here. What happens to the tumbling frequency? No matter where they're going, what? It goes down. It goes down very quickly, within about 0.1 second. All the bacteria are now swimming like this. So they used to be like this, and now they're like this. So that's the response. And then something happens. They start tumbling again. So they realize that they've been fooled. Say, hey, things aren't getting better. Things are just the same. They get restless again, and they start tumbling. they start tumbling, they start tumbling, they start tumbling, they start tumbling, and they reach the same tumbling frequency as before. So, although attractant is present, they adapt exactly back to their original tumbling frequency. This property is called exact adaptation. The time it takes you to adapt is called the adaptation time, tau. And tau can be between seconds to minutes, depending on the size of this attractant step. You put it saturating attractant, let's say 1 millimolar aspartate. They can be stop tumbling for 10 minutes and then adapt. And if you put a tiny amount, 1 micromolar, after a few seconds, they adapt. Now, this adaptation is very common in sensory systems across biology. For instance, all our own eyes, our visual system. So imagine that you're outside in the bright sun and you go into a dark room. So at first you can't see much, but after a while your eyes adapt and you can start seeing small changes in contrast. Right? If you, also the hearing system, if there's a hiss like this in the room, you hear it, it bothers you, and after a while you stop hearing it. Or the sense of smell. If you bake a delicious cheesecake and you will go home, you walk into, you say, what a great smell. And after a while you stop smelling it. So the sensory systems generally, not all of them, not pain, not heartbreak, no, heartbreak, yes, has adaptation. So bacterial chemotaxis too. So it's a great place to understand this, why it is and how it happens. So this exact adaptation. The output returns to a baseline value in the presence of constant input. So the steady state output is independent. Let's take a nice deep sigh of relief on input. But there's still a response. Now, the system we discussed last time, EMS, VOMP, R, all the signaling systems, didn't have adaptation. You put in an input, they respond and stay that way. So that's a lot of signaling systems are like that. But a lot of sensory systems do have exact adaptation. You put an input, they respond, and then they adapt. And that's important for them. Yeah.
Just a technical question. The tau, the adaptation time,
Yeah.
is till it starts rising or until it reaches the final equilibration?
How do you define the adaptation time tau?
Yes.
Yeah, so let's define it as going back to 50% of the baseline. Baseline, I say, the steady state. This is the baseline. Tumbling frequency we'll call F. This is F0, baseline, and you go back to F0. And so we'll deal with this curve and try to understand this curve. We'll try to build the circuit that gives you this computation, you can say. It's a little bit like a derivative. So you have a step and you get a pulse. A little bit like taking the differential over time of the signal. That's what the bacteria do. That's how they compare. That's how they compare now to the past. And okay. And the bottom line is going to be that this property of the exact adaptation, this property of the exact adaptation will be robust to all the protein levels in the circuit. So, but we'll see that in a second. But I'm just giving you the answer so that you can relax. This is the step experiment. Response, adaptation time, precise adaptation. Okay, we're doing very well. So I guess I can tell you another little thing about the bacteria before we look inside of them. Or maybe I should say, why is this exact adaptation important at all? Because it allows you to remain sensitive over this very wide dynamic range. So the thing is, if I give a step, I'm going to plot it here. If this is attractant, and I give a step, attractant, and this is tumbling frequency, I have response and adaptation. If I give another step, again, I have response and adaptation. If I go down, it's like repellent. I have response and adaptation. See? So this keeps me, the tumbling frequency, the search I do over space, at a nice intermediate tumbling frequency. And if I put a lot of attractant, I have a response. And if I put just a little bit of attractant, I can still respond to it. Until the receptors get totally saturated and they can't bind any. Hello, nice deep sigh of relief. Welcome. Can't bind any more attractant molecules. So this is important for, it's a gain control, keeping you also in your visual system. The amount of light, the difference between a starry night and full sunlight is something like 10 order of magnitudes in photon flux, and we can still see not too small relative differences in contrast because our eyes constantly do this adaptation. And also cameras do it. Your cell phone camera does it. So. Okay. We're going to... I want to tell you about the individuality. When you look at the microscope at these bacteria, you see that bacteria have individual character. Some are nervous. Some are relaxed. The nervous ones have low baseline, have high baseline tumbling frequency. These ones have low baseline tumbling frequency, so they tumble more or less per second. These ones happen to have also a short adaptation time and a long adaptation time if you add attractant. This was discovered in 1976 by Spudich, Koshland. Individuality. This character lasts for a whole cell generation. And also the daughters are a little bit correlated. So if I plot here 1 over tau and F0 for different individual bacteria, tau goes like 1 over F0, and this is for different individual cells. So that's another thing we can try to explain. So this is the average over the population, but each cell has a different baseline, a little bit different baseline, a different adaptation time. But each bacterium, every single individual bacteria has exact adaptation to its own baseline. So if a bacteria is a little bit more tumbly, it'll go back to its own baseline. So the exact adaptation is something here that's constant between cells. Yeah.
You said that you have also negative chemotaxis because the repellent is both of them. If you just sum together the repellent part and the attractive part.
So the question is, what happens if you now add multiple signals? And this is actually an area of constant of current research, and we recently discovered, based on theoretical grounds, that what you do when you have two relative, so you compute the relative change here of one input and the relative change of the other input, and you multiply them, and that's the relative change the bacteria perceives. So if you have a twice increase in the repellent and a twice increase in the attractant, it does nothing. That's what we predict based on.
It doesn't calculate the expectancy or whatever if the repellent is twice as hard as the attraction.
It can have. The question is, what if the repellent is twice as dangerous?
Yes.
So it can have some weighting. Depends if it's the same receptor or not the same receptor. And you can have it, in that case, what you do is you look at the fold change of it's a log linear. It's like a product of fold changes to certain powers, and the powers can be different for different chemicals. So that gives you a bigger weight for, let's say, repellent or attractant. But you can simultaneously increase something and decrease something and get zero response. But then you can, there's a lot of things to discover. So you take the bacteria and you put two sources here and here. Where do they choose to go? Or all these kind of experiments, which are very interesting to think about. But we won't get into them. Yeah, I see another question.
How is that advantageous to bacteria? I would expect that a repellent would be dominant over an attractant because you don't want to die more than you want to eat.
So what you're talking about is loss aversion. It's worse to lose than to win. It's worse to die than to eat.
Yeah.
So you see, we're getting into these deep existential questions. Because bacteria, just like us, is a living organism. It's faced with the same essentials of life and death. And so I like these questions. I think they generate a lot of scientific hypotheses. And keep thinking about them. Okay. We're now going to take a look inside the cell. It's just a way of saying, I don't know the answer. So, but you can. You're the next generation of system biologists, right? There's plenty of work to do. I'd like to look inside the cell now at the circuit that does this computation, this derivative kind of function. All right? So we're going to zoom in inside the cell. The chemotaxis circuit is made of 6 kinds of proteins. And some of them you're already familiar with the idea from the last lecture. So I'm going to draw the cigar-shaped cell. See how big it is now? And on one side of it, we have the receptor. So E. coli has 5 different kinds of receptors. Each one can sense different kinds of chemicals. One can sense oxygen. The aspartate one can also sense ribose, etc. And they're actually sitting, E. coli has a cell wall and an inner membrane. They're sitting in the space between the cell wall and the inner membrane. These are details that are not so important for us right now. Attractant binds the receptor with millisecond time scale. So the receptor can integrate and sense the concentration of attractant. And inside, this receptor is connected to a protein kinase. So this whole thing, called key A, this whole thing here, we'll call it X. This is the receptor. And the attractant binding makes it less active as a kinase. Unlike previously where the signal made it more active as a kinase, here it makes it less active as a kinase. And this kinase, what does it do? It takes the response regulator Y, key Y, and phosphorylates it. So you know about phosphorylation now, adding the phosphoryl group. But the thing is, this is not a transcription factor, because in chemotaxis we can't work with transcription factors because they're so slow. If we work with transcription factors, it'll take us an hour. But we want 0.1 second response. No way to make new proteins in 0.1 second. So we can't make new proteins. You have to rely on modifying existing proteins, signal transduction. And that's why everything here will be fast, signal transduction. And this Y phosphate clicks into conformation. It diffuses through the cell, enriches the motor, and binds the motor, and increases the probability of clockwise, which means increases tumbling frequency. Yeah. So tumbling frequency our output, right, increases with Y phosphate. This kinase activity here decreases with attractant. So attractant binds, there's less of this Y phosphate, less tumbling, and we get this response. Now you might ask, 0.1 second, you said the response. How can it reach from here to here in 0.1 second? So that's the time scale of diffusion across 1 micron happens in about 0.1 second. Again, in water it will be 100 times faster, but in 100 times bigger diffusion constant. But in bacteria with proteins that are like this, their diffusion constant is about 10 microns squared per second. That's what you get. Okay. This is the kinase X kinase activity as a function of attractant. Now we need to reset. So you remember in the last circuit, X was also the phosphatase. Here there's a special phosphatase called key Z that does the dephosphorylation reaction. Again, working at around 0.1 second. That's the time scale of resetting. So there's constant phosphorylation, dephosphorylation. If attractant binds, there's less phosphorylation. Dephosphorylation stays constant. YP goes down. Therefore, tumbling frequency goes down. And we're down. Now, if I have a lot of attractant around, Y phosphate is low. There's low tumbling frequency. What's the problem? Did we solve the circuit? How does adaptation happen? The attractant is still around. Kinase has low activity. There's low Y phosphate. How does it go back up? How does it go back up? So there's 2 more proteins here in the circuit. So 2 proteins are necessary and sufficient for adaptation. And the way this works is the following. Is this receptor has 5 buttons. It can be chemically modified by adding CH3 groups. It's called methylation. And the more methylated, the higher the activity. Higher its kinase activity. So this is a way to reset. If you bind with attractant and you start methylating, you recover the activity. Okay? So there's 2 chemical modifications here, phosphorylation and methylation, 2 different kinds of chemical modifications. And this is carried out by 2 proteins. Key R, which we'll call R, adds the methyl groups, and key B, which is called B, removes them. And they constantly add so R adds and B removes and gets rid of this methyl groups, adds methyl and removes it. And this is constantly going, even in constant attractant. You add methyl, you remove methyl, you add methyl. So the cell is actually, that's a significant part of the methylations. It wastes the carbon and the CH3 and a lot of energy just to keep this steady state. Now, what happens here? This is time. This is attractant. Goes up. And this is the YP, which is, as we know, is like tumbling frequency. Goes like tumbling frequency. Goes down. The methylation, this is how much methylation there is. Or let's call it XM, methylated receptors. So X goes to XM by key R, and XM gets demethylated by key B. This methylated receptor starts going up. How does that happen? Linger on, you pale blue eyes. All right. Let's take a nice deep sigh of relief. We need to do some math modeling now. Such is life. We need to understand, we don't need to, but we can't live without going up a level and trying to understand these kind of circuits. So just to baked explanation. What happens if attractant goes a little bit down? Receptors become more active. Therefore, B becomes more active. By the way, this receptor phosphorylates B also, so that's how it becomes more active. Because more active, there's more methyl removed. And R always works at a constant rate, I should tell you. No matter what happens, each of these molecules of R adds one methyl group. That's the constant. So methylation goes down to a new steady state. The attractant, when you remove the kinase activity went up when you remove the attractant, so there's a response. But gradually receptors become less and less active, and you return back to steady state. So that's how this thing works. So let's try to model that. Okay. So I'm going to erase massively now. Linger on.
Is Z the phosphatase for PP?
Z is the phosphatase, exactly, good. So actually, it's a two-component system, but it's one of the only ones that has its own phosphatase, where the receptor is not the phosphatase. Unlike what we studied last time, it's a different game here, because this doesn't need to supply an input-output curve that's in steady state. This needs to have exact adaptation in steady state. So different function, different circuits. Receptor, key Y, key Z, methylation, key R, key B. Mutants in key R, key B, by the way, the receptor it's made with about halfway methylation, midpoint, that's cool. Without key R, key B, there's no addition or removal of methylation. So all you have is the response. So when you add attractant, tumbling frequency goes down and stays there. When you take away attractant, it goes back up. So it's really, there's no adaptation. And those cells are miserable at chemotaxis. I mean, they can't reason their way out of a paper bag. So I think I'm going to I think I'm just going to give you the robust model. I'll spare you the decades of mathematical work that made models that have exact adaptation, but they're not robust. That's when you change protein levels, exact adaptation is really fine-tuned. I'm just going to show you the model called that was figured out how this works. This is Barkay-Leibler model. 97 works using a principle called integral feedback that I'm going to explain to you to provide exact adaptation. Okay, and so we need to set up the mathematics to describe our biochemical situation. So I'm going to call the way this works is like this. This is the unmethylated receptor. And key R, which we'll call R, methylates constantly this receptor. And the receptor can be in active and inactive states. So this active state is the one that gives you YP and gives you tumbling. And the equilibrium between these two states has to do with the attractant binding. So you know that if there's attractant binding, you're more likely to be in the inactive state. Right? So this has to do with attractant binding to the receptor. And at the heart of this model is an assumption. So R methylates the receptors at a constant rate, VR. And B demethylates only the active receptor. So, and that's the feedback here that's so clever, is that this demethylating enzyme can sense the activity state of the receptor and demethylate only the active receptor. It doesn't demethylate the inactive receptor. So no arrow here. And so actually, there's two assumptions you can see. R methylates at constant rate. It's basically what's called for those of you who know about Michaelis-Menten enzymes. This is at zero. What's it called? It has so much X0 to methylate that it always finds an X0. So the rate here is just proportional to how much R there is. And B methylates the inactive receptors. So let's just write down this equation for this situation. Only the active receptors. So the total amount of methylated receptor, active and inactive, okay? Total amount of stuff methyl going into here is going to be R times its rate minus B times the active receptor. That's fine. That's how much you can say. This is how much methylation there is on the receptors, and it's accumulating, and it's taken away. But it's taken away at a rate proportional to the out of all the methylated receptors, that fraction that is active. Now that fraction that is active is X total. The
B part.
The B part? Oh, I see what you're saying. Yeah. Yeah, right. So the rates here, this is, the rates here would be, the units here are 1 over time for VR, and this is 1 over time over concentration. So it's methylations per active receptor. Again, this is a bimolecular reaction. This is essentially unimolecular because R always finds its substrate. It's like saturated. It's a saturated enzyme. And that's a good assumption because there's so few R's. But you need both of these assumptions. You see, that R is saturated and works constant rate, and that B only acts on the active receptor. Okay. And how should I say it? I'd like to call this xm plus xm star let's call this M. This is total number of methylated receptor. Okay, so so this is a slow time scale. On a fast time scale, the balance, this is all here, this thing here, this is m. So the balance between the active and inactive is some function of this amount of attractant. So this function here, let's put it, not f because it's called g. So this g of s is a decreasing function. So the idea is that if I add signal, I have less activity, but then m goes up, 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 and I come back. So this is the model. No, let's just analyze it.
And
x0,
it can't be activated.
okay, so x0, what do you mean? It has no activity. That's right. X0, the unmethylated receptor, can't cause white phosphate. And we know that from experiments. If you, for instance, how can you get all the receptors unmethylated? You take away key R, you delete key R, and then there's only key B. So it takes demethylates, and the cells basically stop tumbling. Delta key B mutants never tumble, for example. So we know that. This is unmethylated, inactive. So let's analyze this. So what happens to this? Right. So this is dm dt. Sorry for erasing like that. I know you don't like it, but I'm going to keep it here. So let's see what happens to. Yeah, let's do rate balance plot. So here I'll plot the activity, and here I'll plot M production and removal. So M production this is RVR. So it's constant. It doesn't matter what activity. The removal looks like this. B times its activity times XM star. There's a fixed point, right? Like we did a long time ago. If we have a little bit of less XM star, production is bigger than removal. So the flow is in this direction. And if I am here, removal is bigger than production. The flow is in this direction. So no matter where I start, I'm going to flow to the same fixed point. XM star always flows to the same fixed point. XM star steady state, which is equal to VRR divided by VBB. Okay. So, yeah?
Why do you have a linear function for removal?
That's the way I wrote the model here.
But XM star is not a linear function.
Okay, let's see. So XM star depends on the signal. But it doesn't depend on XM star. Okay, that's right. That's the second equation. Let's see what happens now. Okay, so if we add attractant S, then according to the signal, XM star, which is M times G of S, goes down. So XM star goes down to this point. And instantly, within 0.1 second. That's the response. But then what happens? Methylation starts rising. Methylation. And this M increases to cancel out this G of S. And because of the way this equation is written, you flow back to the same XM star, which is also provides, of course, the steady state tumbling frequency, because the active receptor is the ones that phosphorylate. So this locked into this equation is that no matter what I do, at steady state, methylation would bring you back to the same activity level. Why is that? Because the feedback loop is closed on the activity level. So if I plot surfactant, input signal. Methylation. Oh, let's put it this way. The output, xm star, which goes into, is of course the tumbling frequency. The output goes down because of this equation. Let's call this equation 1 and equation 2. So this is equation 2. And then we're here. So M production is bigger than M removal. So M starts being produced. M starts being produced. Xm star begins to rise. When does the process stop? When Xm star reaches its steady state, VRR divided by VBB. R is the concentration of key R protein. B is the concentration of key B protein. VR, VB are the rates of adding, removing methyl groups per protein. Yeah, the specific rates. Another way of writing this equation is that the rate of change of methylation is some constant times xm star steady state minus xm star. So so if it's this constant, by the way, is VBB. If I have more than the steady state, then M goes down. If I have less than steady state, M goes up. But the only fixed point, only way to get steady state is when xm star goes to some fixed value. So it's locked in. And this is what's called integral feedback, because the methylation integrates the difference between the activity and its steady state activity. If I put an integral on both sides, methylation is the integral of the error. The error is the difference between the activity now and the steady state activity. That's called integral feedback. And because it's so powerful, because if the error is large, the integral keeps accumulating it until it drives the system back to its steady state level. I'm going to give you an analogy in a second. We turned on the air conditioning here. How's the temperature for you? Somebody asked me to turn it on. Is it too hot? Too hot? Should we increase the air conditioning? Yeah? How many people want to increase the air conditioning? How many people want to keep it the way it is? Okay, so it's a majority. But I'm not only asking because of your comfort, which I care about, but also because how does this air conditioning work? When we set a temperature, how do we know the air conditioner gets to that temperature? The answer is integral feedback. It's the same thing. I'm going to show you in a second. Same thing that evolution discovered 1 billion years ago, and geometry discovered in the 1920s. A very robust way to get adaptation. But before I do that, I want to ask you guys a question, okay? To think about for a second. And here's the question. Think about for a second. I'm going to ask you the question. Question. Is the steady state tumbling frequency robust to protein levels. 1, no. 2, yes. 3, I don't know. 4, depends on parameters. Okay, think about this for a sec. Don't answer me, but I want to make a vote. Okay, so just think about it for a second. And of course, I'm asking this. It might be weird that I'm asking you to vote, but it's in your own best intent, both best interests, because this technique has been proven by the physicist in Harvard called Mazur to increase learning. Okay, so bear with me. So, how many people think 1? It's okay to be wrong here. How many people think 1? 1, 2, 3, 4. How many people think 2? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. How many people think 3? 1. Don't be ashamed, because you're in a course. You're not supposed to know. 1, 2, 2. And how many people think 4? 1, 2, 3, 4, 5. Okay. Now, find someone next to you that, it's best if someone from a different field. So how many people of you are biologists here? Raise up. Now, the ones who aren't biologists, find a biologist sitting next to you and turn and discuss this question. Keep your hands up for a second. Okay, someone's not a biologist, find a biologist. I want you to form pairs and discuss this question. Nobody stays alone. תשאל את הביולוג שיושב לידך. What do you think the protein levels are? Which proteins am I talking about?
I guess R&B.
R&B, yeah. Okay, let's revote. How many people? How many people think 1? 1, 2, 3, 4, 5. How many people think 2? 1, 2, 3, 4, 5, 6, 7. How many people think 3? 1, 2, 3, 4, 5, 6, 7. How many people think 4? 1, 2, 3, 4, 5, 6. Okay. Okay, things improved, but it's not absolute. Like in the previous 2 times, it was a big improvement, talking to each other. Here it's also an improvement, but so this is the correct answer. So let's just understand. Who wants to explain why? Who wants to explain why? Yeah, come.
I have to actually stand?
No, it's fine if you say. Just say your name, please.
My name is
Aryeh, which lab?
in one of those labs.
Okay, great. So first, a big round of applause for volunteering. And you get a big round of applause no matter. So why is it not robust?
Well, we see that both R and B, which are protein levels,
Correct.
They determine the tumbling frequency.
Correct.
So if they change, then we're not robust. And there are no arrows going into R or B, so they don't change.
Good. Round of applause. Yefé. Correct. I agree. So in this circuit, the steady state tumbling frequency it's not robust. So if you have more R, a cell with more R has a higher steady state tumbling frequency. It has a response. It also sometimes depends on the model, changes its adaptation time, not in this simple model, but it returns to the same steady state, to its own higher steady state. So exact adaptation is robust, but steady state tumbling frequency is not. That explains why is it that cells have their individual characters. Why is it cells, some of them are nervous and some of them are relaxed? In fact, experimentalists have narrowed this down to primarily to fluctuations in key R, the least abundant protein, about 100, therefore with the largest relative changes from cell to cell. The ones with high key R tumble more, the ones with low key R tumble less. It also has to do with their adaptation time. It explains the correlation I talked about in individual cells between adaptation time and tumbling frequency. But each of those individual cells has exact adaptation to its own baseline level. I just want to tell you guys, I'm going to say it very quickly, for you guys who know about eukaryotic signal transduction, in the MAP kinase cascade, there's this protein called ERK2. If you don't understand what I'm saying, don't worry. It goes into the nucleus when there's growth factor signaling. ERK2 in the nucleus, this is work that Chelina, Cohen, Saidon did it in my lab. This was the first measurement of endogenously tagged protein, signaling protein in individual cells. Each cell has its own baseline level in the nucleus. When you give a signal, each cell has a response and exact adaptation to its own baseline. That's the way signal transduction works. And so the cells need to know somehow to measure relative changes to their own baseline. So there's circuitry to do that. We'll talk about that later. But this same phenomenon happens in important cancer signal transduction systems, etc., like the MAP kinase cascade. If you want to learn more about that and you're not from biology, you have the partner you just talked to, so ask them later after the class, what's this ERK2? What is he talking about? How many people heard the word ERK2 before? So these are the people you can talk to. It's really interesting. Cancer gene. All right. So I said this integral feedback is also operating in the air conditioning here. And that's the way engineers build robust control. How many people here heard of PID control? Okay, just a few. This is called proportional integral differential feedback. I'm going to erase now, okay, guys? Any objection? Okay. And integral feedback turns out to be at play in a huge range of biological situations where we want to reach a robust, nice, steady state. Integral feedback is a big principle. So let's talk about the heater. Heater uses integral feedback. So we have the temperature. Okay, the temperature. How does temperature change with time? Well, there's the power to the heater or air conditioner, and there's some constant here. So the more power, the more temperature increases, right? Power to the heater, power to the heater. And maybe I open a window. So this is leaking out the window, right? And maybe I add here another, let's say, heat sources. Let's say all the people in the room, each one has 100 watts, you know? But we want to get the temperature to the desired steady state. So we want the T steady state will be something that we set independent, robust, to all parameters. A, which is how good the heater performance. B, the window size. This is basically the thermal coupling to the outside. S, the other sources. I want, no matter what I do, I open the window, I open the fridge, I put more people in the room, the heater gets old, okay? And the same amount of power stops giving you a good temperature. I still want to reach the same temperature. How do I do it? Huh? You increase the power. You need to increase the power. So there's a feedback loop, right? But the question is, what feedback loop? And the way to do that is to put in a thermostat. So you have the heater. This is the temperature. Here's the thermostat that compares the temperature to the desired temperature, which I set this T0 with my knob. This goes through the integral and changes the power to the heater. So what I want to do is change the power in the following way, this way. If my temperature is below the desired temperature, I increase the power. I start increasing the power. It's not that P is equal to this, it's the rate of change of P that's equal to this. If T is too high, I decrease the power. Now, how do I know that this is robust? How do I know that this is robust and reaches T0? No matter, irrespective of A, B, S. What's the only possible fixed point in this system? Is T equals T0. It's exactly the same curve there. I have the rate of adding power is K T0. And the rate of removing power is K T. And their only intersection is where T equals T0. That's the way it works here. K T0 is adding, add power. K T is remove power. This is a constant that I fixed, right? I can change it, of course. That's just like the steady state tumbling frequency can change if I change the protein levels. I can change it. And you see, this fixed point doesn't depend on A, B, S, or K, by the way. K just changes how quickly I get there. That's called integral feedback. And bacterial chemotaxis implements integral feedback. What's the integrator? Methylation. You start accumulating methylation if your activity is different from the set point. This link between, so Barkai Leibler, I was a postdoc when Nama Barkai was a postdoc too. And so I came after Nama Barkai and I did the experiments to show that this model predictions work. But it was really amazing. She took 100 papers and she turned them into this model. Afterwards, an engineer called John Doyle pointed out that it maps to integral feedback like that. And now we have this concept, integral feedback. And the same circuit, you can say, occurs in hormone homeostasis. You give birth, you have less calcium because of placenta. There's a hormone that starts making more calcium. Otherwise, you die. And you have an integral feedback loop, et cetera, et cetera, et cetera. And there's found in many, many different systems. And that's the way, if you want to reach a steady state that's robust, that's the way you do it. So now our theorists already know what to look for in the biological mechanism. But this work was actually the first molecular mechanism for robustness ever. The thing I told you about yesterday was last time with the bifunctional kinase phosphatase, that came after. But the concept of robustness in this modern form, you can say, derives from this Barkai-Leibler model. And people thought about robustness and homeostasis. Homeostasis means keeping steady state a long time ago, like Waddington in development and Katzer in metabolism, and there's experiments to show it, but they didn't know the molecular mechanisms because there wasn't enough biology known. But now that there is, we can look in detail at how this works. And it really depends on key B only demethylating the active receptor, not the inactive receptor, etc. And key R working in saturation. So it depends on weird biochemical details, but they make sense if you understand the robustness principle. And then just to tell you, the experiments, the kind of experiments I did, what I did was let's say I removed key R gene. I removed the key R gene from bacteria. So what happened to those bacteria? They had, how do they swim, do you think? Key R is the one that adds the methylation. It's always unmethylated. Is it always inactive? It moves straight like this. And then I added key R under control of a sugar promoter. So when I add something like a sugar called IPTG, the cells can't eat it. It makes key R, so I can control the amount of protein, key R. And I had cells with low key R, high key R. And I measured this curve for them using I put bacteria in 2 dimensions. I put a drop of bacteria between 2 slides, they covered with a slide, and they were like in this two-dimensional situation, and they swam around. So I could do image analysis of individual cells, and I could add that attractant and see the cells start swimming like this, and then like this, and measure this curve. And when I increased KR, steady-state tumbling frequency increased, adaptation time decreased, but they came back to their own steady state. And that was the way to test the predictions of this model that you can see. Because other models around were not robust. You get exact adaptation, but if you change protein levels, you adapt below or above. So that was pretty exciting. That's how I learned biology. I was a physics PhD student. That was my postdoc. So I was really at the right place at the right time. With this opening in biology. Nam Aberkai came a year before me. Right, so everything I told you is in the book, Introduction to Systems Biology. How many of you have took a look at this yet? Okay, more than half. That's good. And I think this is a good place to stop.